I'm Greta.
I'm Augie.
And our study is on the effect of SAD light treatment on dSERT gene expression, motor function, and aggression in Drosophila melanogaster. SAD stands for seasonal affective disorder, and it's a form of a major depressive disorder that people develop during the fall and winter months with lower light levels generally in the northern hemisphere. And SAD is linked to a downregulation of serotonin, which is a neurotransmitter, which also regulates aggressive behavior and motor function. The question we are trying to answer in our study is, does light therapy increase serotonin levels? And can treatment during development affect adult behavior? In our study, we measured three things. We measured dSERT gene expression. dSERT stands for Drosophila serotonin transporter. And so even though we weren't directly measuring serotonin production in fruit flies, we were measuring a gene that should tell us what our serotonin expression looks like, because it's essentially telling us how many serotonin is being received at the neurons. We also measured or performed a larval mechanosensation assay, which tells us what the motor function was in our larva, and that also should directly correspond to serotonin production. And then in our adult flies, we performed a male aggression assay, so we measured aggression in the adult males. And we predicted that light therapy will correspond to an upregulation of dSERT, increased motor function, and decreased aggression. And our hypothesis is that light therapy increases serotonin production during development, thereby resulting in a decrease in aggressive behaviors in adults.
All right, so on to our methods. We cultured our Drosophila melanogaster fruit flies in hydrated potato media, which is just standard growth media. We did this for 3 days. We allowed the parents to breed for 3 days in order to produce enough larvae. And then we took those cultures and we put them in a light-free environment. We put them in a black box for 3 more days. And during this time, we took the tubes out of the black box that were our treatment group, and we exposed them to a SAD light for 20 minutes each of those 3 days. We synthesized our primer for our dSERT gene using fly-based data and then using the IDT DNA website, which allowed us to create primers for our dSERT gene that were effective and wouldn't fall apart when we put them under quantitative PCR conditions. And so we had to tweak them a little bit, but we managed to get the primers to work, and we managed to amplify our dSERT gene. Our RNA extraction was done on the L3 instar larvae from both our control and our treatment groups. It was used to extract total RNA from these larvae, and the L3 instar larvae is the last larval stage before the flies become pupa. The L3 instar larvae have they're bigger, and so we can get more RNA out of them, so we can get better results. We also did the mechanosensation on these third instar larvae. We did that using a stereoscope and a paint bristle. What we did is we looked at them and tickled their sides, and if they curled around it or they reacted to that tickle in some way, that was a success. If they didn't react at all, then it was a failure. The microsensation assay is somewhat subject to basically problems, and it could be a source of error, which we'll talk about later. The aggression assay was conducted on adult males only. So after we had treated our flies, we killed off all of the parents, and we allowed those treated larvae to hatch. We then isolated all of the male adults from both the treatment and the control group. Following that, we took the male adults and put them into their own little chambers for 24 hours. And then we dumped 2 of those adult males into a small petri dish, and we allowed them to fight. We called it the Thunderdome. And the fly Thunderdome, we recorded it, and then we scored them based on 3 different things, 3 different aggressive behaviors that are outlined in the aggression assay that we've referenced. One was that facing each other was 1 point, a wing flip, which is aggressive behavior in adult males, was 2 points, and then a charge, which is like a really aggressive behavior where a fly will actually go after another one. We scored that with 5 points.
And we did that 10 times.
And we did that 10 times. So we had a total of 20 flies fight for each group, the treatment and the control group. The reverse transcription was done. cDNA was synthesized using a revertate RT enzyme from our total RNA that we had extracted from our L3 instar larvae. We then took this cDNA and we ran quantitative PCR in it, which is that 2x power up cyber mix, which is a master mix. And then we used our primer to target our dSERT sequence, and we amplified it and then compared its expression to the GAPDH gene, which allowed us to normalize our gene expression, which can then give us an idea of its relative regulation between the control group and the light treatment group. So moving on to our data and results, figure 1 is showing preliminary dSERT gene RT-qPCR data. This data is preliminary because we were only able to do 1 round of experiments, 1 RT-qPCR round, due to the COVID-19 disaster. And as you can see, our p-value there isn't very good. We don't have any statistically significant results between the two, but the light treatment dSERT expression is approximately 1.4 times greater than the control group, which means that our light treatment did in fact show like a upregulation of our dSERT gene as compared to the control. But this data is not statistically significant because we weren't able to do enough results. We posit, although, that if we were able to do a few more rounds of RT-qPCR, we would be able to get significant results.
So even though our results from our RT-qPCR aren't conclusive, our mechanosensation assay results do actually really strongly support our RT-qPCR data results, because we were able to actually complete enough rounds of that to have a really wonderful p-value, which you can see down in the table at the bottom. And so basically what these results tell us is that the light treatment group had much higher success rate, and so they showed significantly improved motor function compared to the control group. And as we discussed earlier, motor function should be positively correlated to serotonin expression, and so we would expect to see increased motor function in the light treatment group if we were also getting upregulation of serotonin or of the dSERT as well in the light treatment group.
So figure 3 is more preliminary data. It's preliminary aggression assay data, and the reason being is we were only able to do 1 round of experiments and one round of assays. This bar graph shows the average aggression scores for the adult male flies in the treatment and the control groups. It's actually the opposite of what we were expecting. The light treatment group was more aggressive than the control group, but we cannot statistically prove that that is a difference, that there are differences between the two groups with a really kind of bad p-value in table 1. We would posit that we might receive different results if we were able to do more rounds of the aggression assay. This was scored again by using that aggression parameters that we had outlined in the methods section. And as you can see, the light treatment the results are exactly the opposite of what we were hoping to see or what we had hypothesized.
So what we were able to conclude is that light therapy appears to increase dSERT expression, which should correspond to serotonin production. And again, this conclusion was supported by mechanosensation results, which also support an increase in dSERT expression under light therapy treatment. Our aggression assay results, we can't I mean, obviously they're not conclusive, but they might suggest that treatment during development doesn't affect adult behavior at all. And our sources of error include subjectivity of assays and small sample size in our aggression assay, and then again, too few repetitions of RT-qPCR and the aggression assay. In the future, we would like to complete more repetitions of the RT-qPCR and of the aggression assay.
So there in our reference section, we've referenced the method that we use for quantifying aggression in male fruit flies. And we'd also like to thank Dr. Otto Hitt for making this lab experience a possibility, especially with the COVID-19 pandemic happening right in the smack dab center of it.
It was really wonderful. After we were no longer able to meet for lab, she made sure that we could come in on our own and complete the behavioral assays, even though we could no longer complete the RT-qPCR.
And also, we would like to commend the Carroll Biology Department for enabling an authentic intracurriculum research experience, because we appreciate it and we love what's happening.
Thank you.
Thank you.